to poke a little bit at the, you mentioned AI, right? And this is now we're kind of, in my line of work. And I find this really interesting, and I just think it's an interesting connection to draw, right? So there's a big problem that people have thrown a lot of money at, and that's this problem of protein folding. Like, how do we figure out how a protein folds? And that's kind of how we understand stuff about our bodies and all that, right? And so there's this huge effort going on for quite a few years to calculate, smart computers and really fast supercomputers and stuff to calculate how those things fold up. It's this huge problem. It takes months and months of supercomputer time and all that. And then a few years ago, really just a couple of years ago, people figured out that you could train a machine learning algorithm to not calculate how a protein's going to fold up, but guess. And it turned out that machine learning was so good at guessing that the algos who had inside, they weren't calculating anything. They were just making what we call a swag, right? A sophisticated wild ass guess. And they were better at guessing how those things would fold up than the supercomputers were at calculating how they were going to fold up. So I'm offering that example because we hear AI a lot, and it's become a marketing thing, right? But this is a similar sort of a problem, right? How does this long sequence of DNA translate into how well I'm going to process vitamin D? And we probably still can't calculate that, but what we can do is expose correlations, and we can train machine learning algorithms to guess, with fairly high accuracy how that's going to work out. Correct me if I'm wrong. I think that's what you guys are doing.
Yeah, to a large extent, we are helping with the proteins, and actually, especially on the neurology side, there's so within the cells and the hairs here, deep embedded in these hairs, you've got receptors. And
told you we were going to geek out.
We're going to geek out. You've got these receptors, and these receptors will actually, if your cell membrane is actually two walls, and you've got this protein strand that most of your receptors on the outside of each neuron, they're 7 strands, and they wrap around, and they'll stick out there. And this one receptor, 1,000 types of receptors, grabs nothing but dopamine. Right? This other one grabs, GABA. But I'm going to vector in on dopamine because one of the things that I'm actually doing my thesis paper for my last class at Harvard. So I'm doing an MSc in neurology with Harvard right now. I got bored over COVID, but anyway, for PTSD, one of the problems that they have is someone has it within the hypothalamus within the brain. There's not enough of a certain type of neuron that fires neurotransmitters out that calm down dopamine receptors. And because there aren't enough, memories cannot fully properly process when someone has PTSD. They get stuck there. There's not enough of these interneurons that can calm down kind of hyperactive thinking, if you will. And that's why their images, their memories, which cannot properly process, seem so vivid, seem so strong, and are just so possessing. And one of the things that people are looking at is the ability to genetically modify the receptors, or dopamine receptors, within a certain area called the dentate gyrus within that part of the brain, that actually can turn off a lot of the dopamine receptors. So we may not have the neurons, we may not be able to generate the neurons that can actually stop it, or can actually kind of depress the dopamine reactions, but maybe we can make the surrounding cells less sensitive to dopamine and cut down the reactions right there.